Hello, welcome back. We are on to the second topic in chapter 4, talking about staining. So in your lab kits, you guys are going to be doing some simple stains and negative stains and gram staining, I believe, are the activities from your kit. So we're going to talk a little bit about how your textbook addresses some of those topics. So when you first start getting ready to make a stain of bacteria, we have to do what's called a smear. So it's kind of a fun word to say, but you're making a bacterial smear. So you do this by taking a sample of your bacteria and you put it on your clean microscope slide. Now, sometimes your cultures are going to come from a broth and sometimes they're going to come from an agar. So I haven't worked through how you guys are going to do it yet. I'm recording these before term actually starts. But if you take your culture from a broth, you typically just need a loop full of bacteria and you smear it on your slide, kind of like what we see here, right? So it looks like a little smear of liquid. And then you let that air dry. And so what you should end up with is a cloudy haze of where the bacteria culture air dried. If you can't see a cloudy haze on your slide, then just add another loopful, kind of pile it on there, just smear it around again about the size of a quarter or a nickel on your slide, let that air dry. So keep layering your bacteria on there from a broth culture until you can see kind of a hazy circle of where you've placed the bacteria. Once you get that nice milky haze on the smear, then you need to heat fix it. So you're going to pass that food through the flame. Just take your slide here. This is my, let me see. If this is my flame, take your slide and just go to like 1, 2, 3. Well, you guys are just using a candle, so maybe 4 or 5. You're not going to cook it. You don't want to cook the bacteria. You just want to kill them and stick them to the slide. So when you do put your stain and your rinsing stages, you're not going to wash the bacteria off of your slide. If you end up making a slide, a smear from an agar, like if you're going to pick a little bit of bacteria off of a petri dish, you need to add a tiny little drop of water, and then you're going to mix your bacteria from the agar into that water to make your smear. And in both cases, you're wanting to end up with a cloudy haze of bacteria, and that's going to make sure when you stain, you'll actually have something to look at. So whether you're taking your bacteria from a broth or taking it from agar, you want to make a nice kind of milky, hazy looking smear, air dry, heat fix, and then you're ready to move on to whichever staining technique you're going to do. That's for simple stain and gram stain. Negative stain is a little bit different, so make sure you're following the instructions whenever you get to all of those staining steps. The first type of stain we're going to do or talk about is called a simple stain. So in this slide, this is a picture of bacteria under a microscope without a stain. Can't really see much, right? So there's the whole point of why we want to stain bacteria, because they would be pretty much invisible otherwise. They don't have their own inherent color. So when we add a stain, look at how much more information we can gather about our bacteria once we make the smear, air dry, heat fix, add the stain, rinse the stain, and then we can take a look at it under the microscope. A simple stain just means you're using one dye. You created your smear, you put your dye on there for maybe like 60 seconds, 30 seconds, 60 seconds, whatever it is for that particular dye. Then you rinse it off with distilled water and blot dry with paper towel. You don't dry like this because you're going to scrape all your bacteria off. So straight down, straight up, and that's how you're going to blot dry your slide. And then it's ready to go under the microscope. You do not need a cover slip when you're doing these bacterial stains. So a simple stain, just one dye, 30 to 60 seconds, rinse, blot dry, you're good to go. Gram stain's a little bit more complex. It's called a differential stain. So differential stains use 2 or more dyes. Okay. So in a gram stain, you're going to prepare your smear just like you would anywhere else. You're going to get your bacteria, get your little cloudy haze, air dry, heat fix, and then the first step is crystal violet for 1 minute. Let it sit, and you're going to rinse with distilled water. You do not blot dry in between these steps. Okay, so everybody gets stained purple with the crystal violet. Then you're going to do iodine for 1 minute. This is what's called a mordant. Iodine helps make the crystal violet crystals stay really good in those big gram-positive cell walls. Again, rinse with distilled water. You do not blot dry, just rinse, put the iodine on the wet slide, let that sit for a minute, rinse. Then you're going to do what's called the decolorizing step, and this is probably the most tricky step, is you're going to hold your slide at an angle like this over your staining tray or sink or whatever. So here's your little catching container. You're going to drip your ethanol onto this angled slide. You don't want to let it sit flat until your runoff, it's going to start running off purple, and then you want it to run off until it's almost like a clearish pale pink. It says until it's clear. Oh, it just says 30 to 60 seconds. But I always advise my students, drop your ethanol. This is ethanol. Drip that ethanol over your smear until you can see the crystal violet kind of washing away until it's kind of a pale pink. And then so what you're doing is you're decolorizing the gram negatives. The gram positives are going to hold tight onto that crystal violet. You're getting all the crystal violet off of the gram negatives. Rinse with distilled water, right? So that's really important. You do not want the ethanol to stay on there because that could affect your outcome. And then you do safranin for 1 minute. So safranin is the pink dye. So it's all of those decolorized gram negatives are going to pick up the safranin and appear pink. So then in this mixed slide, you're going to have both the gram positive purples and the gram negative pinks. You'll rinse with distilled water, blot dry, and voila. It's really cool to do these mixed slides. You follow all the same steps for the one slide, and you can get different outcomes based on that cell structure, right? The thick peptidoglycan for gram positives, the thin peptidoglycan and outer membrane for the gram negatives. Okay, and so some of these we are not going to do because they just weren't included in the kit, but in a typical term, we would have students also do what's called an acid-fast stain. So acid-fast stains are primarily for bacteria that have thick cell walls that are filled with what's called mycolic acid. Mycolic acid it's really waxy. Wax is a type of lipid, and so it resists watery things, and most dyes are water soluble. And so, saw a fly, make sure it wasn't a bee. So you have to use something besides these water soluble stains. And so in an acid fast, it's a differential stain. So you make your smear like you normally would. What you'll do is you add what's called carbonyl fusion, which is lipid soluble. You usually put it over heat for 5 minutes. It drives that primary stain into these acid fast cells. Then you go and you rinse it with an alcohol acid rinse. So it's ethanol plus hydrochloric acid. And so in organisms that have this thick waxy cell wall, they hold on to that primary stain, but any of the other ones gets decolorized, and then you use your secondary stain, your counter stain, which in this case is methylene blue, and non-acid fast cells kind of look purple, acid fast cells stain pink. The cells that contain mycolic acid are in the group called Mycobacterium, which is the causative agents of leprosy, so Mycobacterium leprae, as well as tuberculosis, which is a huge public health problem in a lot of developing countries. So tuberculosis is caused by bacteria that have this thick waxy cell wall, this mycolic acid, and so the samples have to be stained in this particular way. Otherwise, it'd be hard to determine if they actually have the bacteria in their lung tissue or their sputum. Endospores, we already talked about endospores back in chapter 3. Some cells have that structure, that ability to create those structures, right? Bacillus and Clostridium. So in an endospore stain, you use a primary stain which stains the endospore, which is what's called malachite green. And then we use safranin as a counter stain, which stains the vegetative cells pink. So in a field of cells that have endospores, the endospores will look green, the vegetative cells look pink. Okay? Again, that's something that we cannot do with this kit this summer. And then lastly, we have histological stains, which are stains of tissues, usually of multicellular eukaryotic organisms. But a lot of times these histological stains allow us to see pathogens that could be in our tissues. So in the left, this particular type of stain called Gomori methenamine silver allows us to see fungus infections, if that's not a good call. Fungal infections, kind of all the black kind of squiggly bits, that's hyphae. That is fungal infection of a tissue. And then on the right here, this is called hematoxylin and eosin, which looks, if you've taken A and P, this is your thin skin. So this is going to be like maybe from your arm or something like that. So we can see the epidermis and then the dermis here. And if there's bacteria or fungus or other things, we'd be able to see those show up in these histological stains. Histology is a study of tissues, so it's staining multicellular organisms' tissues. Another type of stain that you will be doing, I think you are going to be doing this with Congo red, which is more of this color here, is called a negative stain. So a negative stain stains the background and typically leaves the cells unstained. So this is a good example of a negative stain. This is what one would have looked like if we're to have an in-class lab. We use a dark stain called nigrosin. It stains the background. The cells remain unstained. It's just kind of another way that we can do that visualization of the bacterial shape and morphology and things. A capsule stain, like I showed you when we were talking about capsules way back in the beginning of chapter 3 in glycocalyces, is at first you stain the background, then you stain the cell, right? And so the cell is kind of this darker pink, and what's left unstained is the capsule. So again, this is a special staining technique to allow you to see the structure of the capsule outside of the cell with the dark background. Okay, so the last structural stain that we'll take a look at is what's called the flagellar stain. So as the name implies, flagellar stain stains flagella for bacteria that have them. It's a really difficult stain to do. It's not one that we would normally do in class. I've tried to do it in the past, but it never really turns out. Flagella are very, very small and very, very delicate. And so to do a proper flagellar stain, you have to add layers and layers of mordant, which kind of build up the structure of the flagella. And then you can add the stain, and then you'll be able to visualize the flagella. So it's not really worth our time in lab to do something. So we talk about it in a lecture. It's kind of cool to see flagellar stain, but I've only seen a very few, and it's just really difficult to do. All right, so that wraps up our section on staining. The next installment for chapter 4 is going to be dealing with classification to wrap up chapter 4. All right, so I will see you next time.